co-authors are Daniil Pashin and Alexander Chupakhin. And our work is based on medical data from our collaborators, which are Federal Neurosurgical Center and Mishalki National Medical Research Center in Novosibirsk, and one from Moscow, which is Burdenko National Medical Research Center for Neurosurgery. Okay, so probably as you know, aneurysms and cerebral aneurysms are one of the main causes of mortality and morbidity in according to World Health Organization, and they are causes of strokes and various brain or mental diseases like Alzheimer's diseases and other dementias. Here on the X-ray angiogram, you can see a huge, giant aneurysm inside the human brain, which is of approximately 7 centimeters in size. And so this can be in human without treatment for a long time, and they are, of course, are prone to sudden rupture. So there are a number of approaches to calculate the risks, to assess the risks of aneurysm rupture, and they are, of course, hydrodynamic ones and the mechanical, and probably the most easier one, the geometry. I mean, not the easier to do, but the one which is how to put it, which one could do at first place. And for example, the physicians or neurosurgeons, of course, they tried first to do some statistical analysis on the size of the aneurysms, such as the ratio of the of various sizes of aneurysms and ratios to the neck size of the aneurysm. And the problem here is also related to the question whether this or that aneurysm should be treated at all, because there are, of course, some risks of post-operative complications, and they are sometimes comparable to the aneurysm rupture. So just a few slides on various approaches. Well, first of all, this is a strictly geometric approach, which is based on Murray's law, and it relates diameters of the vessels on the vessel bifurcation, and well, based on this analysis, scientists try to predict whether there might be aneurysm appearance. I mean, it can grow there or not. And of course, one of the basic, well, one of the first approaches from the engineering science was to analyze hydrodynamics of the flow inside the vessels with aneurysms. And there is a very large number of such works with a very large amount of cases considered and analyzed. And there are also new approaches, for example, like this one, which is based on a Reith number, appeared first or used in knot theories not theory, and this approach was generalized to surfaces, and with such an approach the authors try to analyze different aneurysm configurations. As for our work, we are considering we will do numerical simulations in the full hydroelastic, like as a we will model as a the system as a hydroelastic system, which is a problem of fluid structure interaction. And here the problem is the main difficulty of this problem is that the flow domain, the boundary of the flow domain is unknown, and this domain, which is an elastic membrane or shell, is provides a counter force and affects fluid flow. So the interfaces or elastic membranes, they were considered from different points of view. There is a theory of energy of the interfaces, or they were considered from the point of view of geometry and differential equations. And I should mention here that the energy of an elastic shell can be described as a series with respect to the thickness of the membrane. And our approach here will be focused on the so-called Wilmer energy, which is, or another term is bending energy, which is which relates to the deformations of the membrane transverse to its or along its normal. So our aim is to assess different types of energies for such a hydroelastic system with applications to the aneurysms, and we will also consider aneurysms with the so-called blebs, which are small sub-aneurysms or secondary aneurysms growing on the main aneurysm. Okay, so we will describe our flow with using Navier-Stokes equations. For membrane, we will, or elastic shell, we will use basic linear elasticity, and these two equations will be coupled through the boundary conditions on a fluid structure interface, which are the equality of velocities and forces on the interface. As for boundary conditions, we can use in a number of cases, we used clinical data, which could be obtained from, for example, from intraoperative measurements. For example, in Mishalkin Medical Center, we used volcanic combo map endovascular measurement system, which has a dual pressure measuring pressure, dual sensor measuring pressure and velocity. This is very thin, tiny sensor placed into the area of interest, and with such a system, one can get velocity and pressure inside the flow. For example, this is this on the left is X-ray angiography, which is used to control the measurement side. On the right, this is the screenshot of a combo map device showing pressure in yellow and velocity in cyan colors. And here we also designed software for real-time acquisition and pre-processing of the data. Here we have flow rate. This is flow rate pressure loops, and here we have some energy loop. No, on the right is velocity pressure loop, and on the left is flow rate and the energy flow rate loops for the flow. And these are these loops in different arteries, with different pathologists. On the left, this is for arteriovenous malformation during the embolization procedure, and in the middle, this is for arteries with arterial aneurysms, and on the right is top figures are top diagrams for veins, and bottom ones are lower ones for sinuses. What's interesting here is that during time, the curve is the point on the curve goes counterclockwise for arteries and clockwise for veins and sinuses. And you can also see different, obviously the shapes of these diagrams is different. Okay, so just a moment. The PowerPoint is just freezed on my computer and won't change the slides. Okay, let me. Is not responding. Okay, let me restart the PowerPoint.
Okay, please do it, because for us it's also we see the same slide, it doesn't change. Have you sent your presentation to the organizers?
Oh, no, that's fine. I think that's it was. I don't know. Yep. Just once again. Here it is. Okay, this should be next slide.
Yes, we see another slide, yes.
Yeah, okay, so as the first example, I would like to show you our simulations related to comparing two approaches, rigid wall approach and elastic wall approaches. Here we, this is a configuration from Federal Neurosurgical Center. We performed 5 cases, 5 calculations with one of them in rigid wall assumption and 5 with elastic wall assumption. And in FSI simulations, we in 2 of them we set patient-specific flow rate at inlet, and for another 2 of them we prescribed harmonic flow inlet conditions. And also we modeled aneurysms in the 2 cases as shell with different mechanical parameters like elasticity coefficient. Okay, so this is the results of the simulations showing from different sides of the aneurysm, the streamlines of the flow for rigid wall assumption and in elastic in configuration with elastic wall. So basically there are almost no changes in the flow, except that the shape of the aneurysm slightly deforms. Well, of course, the deformation are showed in scale. And another, what's interesting was that for that the rigid wall assumption in rigid wall assumption, wall shear stress has maximum at the site of the maximum of the wall shear stress coincides in both cases as both for rigid wall assumption and elastic wall assumption. So, and as neurosurgeon said us that this site was the place of the aneurysm rupture. So if one wants to predict the site of the aneurysm rupture, then probably rigid wall assumption is enough to guess such a location. And sorry, this was another one. Okay, so now the case of multiple cerebral aneurysms, which in this case we compared. The question was how to predict which one of the multiple aneurysms would rupture first. So here we had several cases with multiple aneurysms from 2 to up to 4 aneurysms, and we knew which one of them ruptured. They have different sites on different sides of the brain. And first of all, of course, we compared hydrodynamic parameters such as pressure and velocity of these inside in the flow, but these parameters didn't provide us any good comparison indices. So we calculated mechanical energy dissipated due to viscosity of the flow. This is the integral of the vorticity of the velocity field, omega squared, and also besides this capital W, we calculated total dissipation energy per one period, E, capital E, and average dissipation energy per unit volume of the aneurysm, and also dissipation energy per unit surface of the energy, and the total dissipation energy per unit surface of the aneurysm. So, and after comparing the results, we can see that in first of all, we had 5 cases, and every such quantity was less than others, was less for ruptured aneurysms in most cases, that is at least in 3 of them. And the average dissipation energy per unit volume was less for ruptured aneurysm, which is here in orange, is less than this quantity for other aneurysms, and this quantity was less for ruptured aneurysms in all 5 cases. So this quantity can be used as a predictor for aneurysm rupture. So next step is to consider the surface of the aneurysms, and in this case we computed the energy of a hydroelastic system as a sum of different energies. First energy is elastic energy, which depends on the area of the membrane or shell. The second energy is well, this one relates to the fluid. This is kinetic energy of the fluid, and the next quantity is Wilmer bending energy, which is computed as an integral of the mean curvature of the surface squared. Here, capital E and nu are parameters of the material, and d is a wall thickness. And along that energies, we computed the dissipation energy of the fluid due to viscosity omega squared, integrated over the aneurysm volume. We consider several configurations modeling these modeling fusiform aneurysm, and the aneurysms were modeled as a sphere, and the configurations were with turning vessels by 45, 90, and 135 degrees. And along these configurations, we considered also configurations with growing bleb or secondary aneurysm with the heights of fourth of diameter of the main aneurysm, half of the diameter, and 3/4 of the diameter of the main aneurysm. And to compute to perform the analysis, we designed a computational framework in which the simulations were performed in ANSYS software. We used CFX and mechanical solvers coupled with ANSYS, and the post-processing. For post-processing, the data were exported with the export inside module, and after that there was we used Python software in conjunction with the VTK module, and the curvatures were computed in the MeshLab server. After that, we used again Python module to form the final tables with the results. And so, first of all, the simulation results. These are just streamlines and wall shear stresses and configuration obtained in the simulations considered, a couple of them. And as we can see that the turning vessel results in a complication of the flow inside the aneurysm with the more complex structure of the vortex. And if we have a secondary aneurysm or bleb in some well, initially the flow just goes inside the bleb, and after the bleb growth large enough, and there is a secondary vortex formed inside this bleb. Well, as for wall shear stress analysis, I also would like to mention here that there are two types or two theories actually, as whether the high or low wall shear stress gives growth to the aneurysm. And both of these approaches are supported by some researchers. And so here we can't just rely on WSS analysis. So we analyzed the energy components of the total energy of the system, and here the results showing that well, on the left these are just time series of the energies for vessel without for a straight vessel, and for a vessel turning by 45 degrees, and for different sizes of the bleb. And on the right, the summary diagram showing average elastic energy for different cases. And of course, with the growth of the bleb, the elastic energy increases as the surface of the system increases. But inside each of the group, we have a slight growth of the energy with angle. As angle increases, the elastic energy also increases, which shows that the deformation of the system of the membrane increases. As for bending energy, this is our Wilmer. We considered the Wilmer functional, and again, each group with on average with the larger the bleb, the higher the Wilmer energy, but inside each of the group, this energy, bending energy, slightly decreases. And this is due to the bending energy characterizes the waves on the surface of the vessel. So the more complex flow, then the more smooth or smoother the surface of the vessel. And kinetic energy increases, of course, with the angle of the it increases with the angle of the turning with the turning angle of the vessel, and on average the between the groups with different sizes of the bleb, the energy is more or less the same, kinetic energy. And the viscous energy follows the kinetic energy pattern here. And we compared also the total energy of the system here on graphs with the different colors, shows the sum of different terms. That is, in orange, it's just the elastic energy, and then we added kinetic energy, and finally the bending energy here of the system. As you can see, that the kinetic energy and bending energy is almost of the same order of magnitude, which is more easily seen on these diagrams. They are not from 0 to 1, but they show that the main part of the energy, total energy, is elastic energy, which almost to 100%, and small terms for kinetic energy and bending energy are of the same size. So probably these two are connected, and one energy can be related to another. And finally, we also considered the diagram, velocity pressure diagram inside this system. We calculated velocity and pressure in 5 different sides of the system. And on the right, you can see these diagrams for a real patient. And these are the results which show that in our calculations we obtained very similar forms of these velocity pressure loops. And so to conclude, we constructed a computational framework for analyzing elastic systems, modeling vessels with aneurysms. And in these systems, the kinetic energy of the fluid and the bending energy of the vessel wall are of comparable size, which probably shows that they are related. And in these simulations, we show that velocity pressure diagrams have close resemblance to the ones for real patients' data. Okay, and along the vessel, these diagrams change. So that's all. Thank you.
Thank you very much for this very interesting presentation for such an important topic. I would like to recall our listeners that they can ask questions either in chat or raising hands. So please, we have some time for questions. And I see that Alexei Tokarev has a question. Please, Alexei, go ahead.
Yes, Alexander, could you explain, please, when you first showed that pressure velocity diagrams, you mentioned that the point rotates clockwise or counterclockwise depending on the vessel in aneurysm, in arteries, and at vein it differs. Could you explain why? What is the physics behind it?
Well, we think that the reason behind it is that in the arterial part of the vessel tree or vessel system, the main force is for fluid is the heart, which is like pushing the fluid. But in the venous, in veins or sinuses, the blood is actually how to put it, sucked by heart. I mean that the force moving the fluid is not pushing, but I would say sucking.
So in arteries we have velocity first, pressure second, and in veins we have vice versa. Pressure. No.
And of course, there is also a difference in vessel wall structure. That is, the arteries have muscles pushing blood, and in veins, no, they do not have muscles.
Yes, we have different structure, but I'm interested in the what. Well, could you please show the diagrams which
Yeah, sure.
the slide with diagrams, arteries and velocity and veins.
Just a moment. Here it is.
Yes, here. So in arteries, we have counterclockwise. Yes.
Yes.
Counterclockwise. So velocity increase and then pressure increase. So we have pushing. Yes. So we have pushing pushing blood into the vessel, and then it causes increase of the pressure.
Yes.
I see. And in veins, we have
it's clockwise. Yes.
Clockwise. So pressure increases first. So it is why is the pressure increase and then velocity increase?
Well, I think that this is due to the very that the vein walls are actually more deformable. So when the when we push the blood, how to put it, the diameter of the veins first increases, keeping the oh no, just a moment. Yeah, so well, I'm not a physiologist, so we thought that this is due to the difference in the vessel wall structure. So that is because the veins can, the diameters of the vein can increase. They can keep the pressure actually lower, but here
well, I think that we can discuss this question
the question is
in more detail later.
Yeah. Okay.
Yes, so,
maybe it depends on the muscles that cause veins to push blood, because well, as far as I understand, the large force which push blood is from muscles which around veins. Okay, I suggest we will discuss later. Thank you very much.
Thank you.
And it's certainly should be discussed in more detail. I would like also to ask something about what you have presented. Very interesting results about, in particular, the relation of wall shear stress with the aneurysm rupture. You mentioned in the first part of your presentation that the maximum of wall shear stress is observed exactly at the place of the rupture.
Yeah.
So if it is like that, so can we then formulate the criterion of rupture that wall shear stress should exceed some given value? Is this condition?
No, we didn't do such research. In this case, we just
quite logic. If the rupture occurs exactly at the place of the maximum of wall shear stress, then we can expect that they are related to each other, no? I mean, the rupture and the maximum of the wall shear stress.
Well, probably you are right. That is, but since we had just this one patient with such configuration, well, when we showed the results, they just mentioned, well, not just mentioned, but that they say that the place or site of fracture coincides with the maximum. So
yeah,
probably we can relate that maximum, for example, that there is a certain difference between maximum of the wall shear stress and the average wall shear stress on the average over the aneurysm surface. But to do such research, we need more data, and right now we have only this one case, unfortunately.
Okay, yes, I understand, of course, that this is a complex question, but you can keep in mind this question. I think,
yes,
according to there can be some relation. And also another question related to the energy. You presented this viscous dissipative energy in the case of multiple aneurysms, and you explained us that in the ruptured aneurysms, this viscous dissipative energy has lowest value.
Yes.
Right. So this is also interesting and not exactly clear. Could you maybe explain why it is, why this viscous dissipative energy is low, takes the lowest value in the ruptured aneurysm? What is the relation between this viscous dissipative energy and the aneurysm rupture?
Well, of course, that we may only guess, but our guess, and that we need more complex research to answer this question. Our guess is that the less energy is dissipated inside the aneurysm, the more impact aneurysm wall has from the fluid inside the aneurysm.
Mm-hmm.
Could that be an explanation?
Well, it's difficult for me to say, of course, at Moreover right now, but I think, yes, obviously, according to your result, there is some relation between flow and aneurysm rupture. But it certainly requires some more thinking and more discussions about that. So please. And then you presented other types of energy: elastic energy, kinetic energy, and Wilmore-Bending energy. But do you have some ideas how these other types of energy can be related to aneurysm rupture?
Well, right now, well, this is ongoing research, so we just did some these preliminary simulations in very simplified geometries, and we are planning to, of course, to relate these energies to assess the risks of rupture or similar questions.
Yes, okay. Thank you very much again for this very interesting presentation. It really makes think about that. And there are also, of course, many questions, so I hope we'll return to these questions maybe later in other seminars and conferences. Thank you again. And now we will start our session on cancer modeling. And I will continue chairing the session because, as I mentioned before, Alexander Bratus has some problems with connection. So the first speaker of the session is Maxim Kuznetsov. Maxim, I hope you hear me. So could you please share your screen to prepare your presentation?
Can you hear me, Vitaly?
Yes, we hear you. So please, you can share your screen.
Yes, that's nice because I also have problems with connection. But I hope that it will go okay. So can you see the slide?
Yes, we see your slide. So then we will start our session on cancer modeling. And it's a pleasure for me to introduce our first speaker of the session, Maxim Kuznetsov from Lebedev Physical Institute of the Russian Academy of Sciences and also from People's Friendship University of Russia. And the title of the presentation is Combined Influence of Nutrient Supply Level and Tissue Mechanical Properties on benign tumor growth as revealed by mathematical modeling. Please, Maxim, we are listening to you.
Yes, hello everybody, and thanks for the opportunity to present the results of my work. Okay, the most notable property of cancer cells is their ability for unlimited growth under favorable conditions, which by itself should lead to exponential growth. All cancers except for blood cancers form solid tumors, and in practice their exponential growth manifests itself only at the earliest stage. Experimental studies on the growth of multicellular tumor spheroids in nutrient solutions repeatedly produce S-shaped or sigmoidal growth curves. Several factors were shown to limit the growth of spheroids. One of them is diffusional limitation of nutrient supply from the surrounding solution. It leads to the fact that in a sufficiently large spheroid, only a fraction of cells situated in its rim can obtain enough nutrients to proliferate. Cells located deeper enter a quiescence state, which requires significantly less nutrients to maintain. The radius of a spheroid that has such a structure increases almost linearly with time, and with further growth, tumor cells in the core of the spheroid begin to die under severe nutrient depletion. The flow of the dead material out of the spheroid slows down its growth rate, and spheroid growth saturates when the dead material outflow compensates the ongoing tumor cell proliferation. The growth pattern of a solid tumor grown in a tissue is analogical in case of compact tumor growth. Such growth type is intrinsic to benign and low-stage malignant tumors. Upon tumor progression, its cells develop two main mechanisms to overcome starvation. One of them is induction of angiogenesis, that is the formation of new capillaries in the tumor microenvironment. Angiogenesis by itself is generally considered to not change tumor growth pattern qualitatively. However, transition from avascular to vascular stage expectedly leads to increase of its growth speed at a linear stage and to increase of tumor maximum volume. Another mechanism is invasion of cells into surrounding tissue, which allows them to move away from nutrient-deficient regions and changes tumor growth pattern at a qualitative level. Notably, such tumors don't even have a clear border with normal tissue. That significantly complicates tumor treatment. Therefore, invasion of nearby tissues is a direct indicator of malignant cancer. Another growth-limiting factor, which was underestimated until the end of the 20th century, is stress-induced growth inhibition. Nowadays, it is clear that the rate of tumor cells proliferation depends on the stress exerted by the solid components of a tissue, that is, the cells and the elements of the extracellular matrix. In one of the first works on this subject in 1997, it was shown by growing tumor spheroids in gels of different rigidity that a compressive stress is able to inhibit the proliferation of cells in spheroid. These findings were confirmed and extended by later works from the same group and by other researchers who used different experimental methodologies. Importantly, the proliferation and death of tumor cells by themselves influence the distribution of solid stress within the tumor and surrounding normal tissue. Solid stress can cause organ dysfunction and lead to capillary compression, thus negatively affecting the delivery of drugs to the tumor. Moreover, it has been shown to stimulate malignant tumor phenotype and to induce resistance to chemotherapy. Therefore, mathematical modeling of tumor growth with account of solid stress, which can in particular address these issues, is of significant theoretical and practical interest. In the first works in this area, tumor tissue was considered as a liquid-like medium or an anisotropic linear elastic medium. Despite the relative simplicity of such approaches, their use allowed reproducing some experimental observations on a qualitative level. For example, the above-mentioned effect of reduction of the maximum size of tumor under the increase of externally applied pressure. Also, such approach allows obtaining non-trivial hypotheses as well of qualitative nature. An example is the possibility of reaching the final tumor size in a non-linear manner, non-monotonic manner under certain conditions. Recently, more complex approaches have appeared, adapted from the area of solid mechanics. Such models can yield more realistic reproduction of tumor stress distribution within the tissue and can even provide quantitative predictions, some of which are consistent with experimental results. However, their solution is associated with much greater computational cost, and they are not amenable to analytic investigation like simpler approaches. It should be noted, however, that in general, modeling tumor growth with account of biomechanical properties is not a very popular area. One of the unexplored topics, to my knowledge, is the combined influence of both crucial growth-limiting factors on tumor growth, which are nutrient availability and mechanical stress. So my goal was to create a simple model amenable to analytic investigation and to study this problem. Not surprisingly, such model contains a lot of assumptions at this stage. First of all, it considers spherically symmetrical growth of a non-invasive avascular tumor in normal tissue, so it has clear borders and there are no capillaries inside it. Tumor and normal tissue consist of porous solid matrix with volume fraction C and interstitial fluid that is able to flow through the pores with volume fraction F. Solid phase can be locally produced, for example, by tumor cells proliferation with the use of interstitial fluid and substances dissolved in it as a source of mass. And destruction of solid phase, for example, cell death, happens with its transfer into fluid phase. The rates of these processes depend on the local level of glucose G and on the local solid stress sigma in general case. The choice of glucose as the crucial nutrient is due to its major energetic role in tumor cell metabolism and its indispensability for biosynthesis. For simplicity, it is assumed that solid stress is isotropic, that is, it acts with equal magnitude in all directions. On the assumption that the densities of two phases are equal and constant, this results in the following general equations for dynamics of tissue phases, where IF denotes the velocity of convective motion of interstitial fluid through the pores of the solid phase, and IC denotes the solid phase convective velocity. Further, the tissue is considered to be fully saturated, that is, the two phases constitute its entire volume and it's normalized to unity, so there are no voids. Summing the two equations for phases dynamics yields the following relation for the velocities of tissue phases. And under further assumptions that, firstly, all external forces are negligible, for example, gravity. Secondly, the interstitial fluid flow through the pores is slow enough for the inertial effects to be insignificant. And thirdly, the solid phase is homogeneously permeable. Darcy's law can be used to obtain the rate of volumetric flow of interstitial fluid the following way, where K is the solid phase permeability and mu is the fluid viscosity. P is the fluid pressure. From the porous media theory, in our case, it follows that the gradients of fluid pressure and solid stress can be linked the following simple way. Therefore, this equation for solid phase velocity can be deduced. Of course, in general, the permeability of the solid phase, K, should depend on the volume fraction of solid phase. However, such dependence is neglected in this work for two reasons. Firstly, in this work, only dynamics of cells as solid matrix constituents will be accounted for explicitly. However, the implicit variation of extracellular matrix density will be assumed to significantly influence the value of K. And the second reason will follow from the simulations. It is that in the considered system, the local fraction of tumor cells will vary in rather moderate range. So the full system of equations that accounts for the above-mentioned assumptions is given here. Tumor cells and normal cells are assumed to have equal densities. Tumor cells consume glucose, and they can proliferate and die. The only component of the dynamics of normal cells is their passive convective motion, analogous to that of tumor cells. Note that the equation for the interstitial fluid dynamics does not need to be considered explicitly due to the fact that the sum of the cells in the fluid are equal to unity. The model accounts for two mentioned tumor growth limiting factors, that is, tumor cells can proliferate only under significant level of glucose and significantly low solid stress. Otherwise, they become quiescent unless glucose level is too low. In this case, they die due to nutrient deficiency. Herein, quite simple forms of dependencies of tumor cell proliferation and death rates on glucose level and solid stress are considered. That is, the smooth approximations of the Heaviside step functions. During analytical study, epsilon will be considered to be infinite, therefore, these will be just switch functions. If the glucose level or solid stress pass a certain threshold, cell behavior changes. All parameters of the model are positive, and they were estimated according to the data of various experimental works. Since the vast majority of glucose flows into tissue by diffusion through the walls of capillaries, only this type of transvascular transport is considered in the model, and the capillaries are assumed to rapidly lose their functionality and degrade within the tumor due to their rupture in the result of their displacement and due to chemical reasons. In light of this, and since angiogenesis is not considered in the work, the surface area density of capillaries is simply taken to be proportional to the fraction of normal cells. The coefficient of proportionality being implicitly accounted for in the parameter P. Proliferating tumor cells consume glucose faster than quiescent cells since they use it as a substrate for biosynthesis. Glucose consumption by normal cells is neglected for simplicity. Parameter P was varied to study the influence of nutrient supply level on tumor growth since it varies in different normal tissues depending on their metabolic demand. Solid phase permeability K and fluid viscosity mu are used in the work only together as the fraction K over mu. Therefore, it will be varied referred to as a single parameter of tissue hydraulic conductivity. Its basic value is based on experimental measurements performed on tumors grown in mice. However, this parameter varies significantly between tumors of various cell lines, at least over 4 orders of magnitude. It has been shown to inversely correlate with the tissue content of collagen, which is one of the main components of extracellular matrix. Therefore, its lowest values correspond to tumors derived from fibrous connective tissue and characterized by the abundance of proliferating fibroblasts, the cells that produce the elements of extracellular matrix. The function of solid stress is a smooth function which corresponds to experimental observations at a quantitative level. It assumes that at normal cell fraction, cells interaction yields zero solid stress. If cells are pushed together, the repulsive interaction appears, that is, sigma is greater than zero, and with an increase in the distance between cells, the adhesive interaction manifests itself. At first, it strengthens, and then gradually weakens due to the sequential ruptures of individual intercellular contacts. The stress eventually becomes zero. An important approximation that significantly simplifies analytical tractability is the case of infinite hydraulic conductivity. At this limit, rising infinitesimal gradients of solid stress lead to instantaneous redistribution of tissue phases and equalization of solid stress. Thus, the fractions of cells and fluid in tissue can be only equal to their normal values. The equation of cells velocity degenerates in this case. Instead, a new equation for it can be found by summing up the equations for cells with the count that the derivatives of their sum is now equal to zero. This results in this system of equations. An important question is when this approximation is applicable, and it is to be answered via numerical simulations. But first, let's stay in this limit and study it. The system of equations allows for semi-analytical derived approximations of the tumor growth curves. And an important moment for the tumor growth curve estimation is the moment at which quiescent tumor cells have just begun to appear. The distributions of the variables are shown here. Since non-invasive tumor is considered, there exists a clear border between tumor and dermal tissue, and moreover, the point in space where glucose level is equal to Gp indicates a clear border between proliferating and quiescent cells. The latter, I repeat, have just begun to appear, therefore it is a zero coordinate. And an analogical situation will further be with the border between quiescent and dying cells. Thus, in every moment of time, the solution can be split into several regions, in each of which some terms of the equations for glucose can be omitted or simplified. Importantly, since the tumor growth speed should be significantly lower than the characteristic speed of glucose diffusion within the tissue, quasi-stationary approximation can be used for it in every moment, that is, setting its partial derivative over time to zero. So, glucose profile can be found by the so-called method of stitching of functions. That is, the coefficients of the common solutions of these equations should be picked in such a way that glucose distribution turns into a piecewise function that is continuously differentiable in order to provide the continuity of glucose concentration and its flow. On the left border, zero flux boundary condition was used for glucose, and the initial size of the normal tissue was taken to be infinite for simplicity. Therefore, the right boundary condition for glucose was setting it to unity at infinite coordinate. The tumor radius can be found as the solution yielded by stitching of glucose distributions, and interestingly, this function has limits for both infinitesimal and infinitely large levels of nutrient supply from the normal tissue. Remarkably, for infinitely large normal tissue, the maximum radius of a tumor that consists of only proliferating cells thus cannot fall below a certain threshold under arbitrary small level of nutrient supply from surrounding normal tissue. This is in contrast with the corresponding result for planar geometry. According to the results of the previous work, such limit would be zero in this case. From the beginning of tumor growth up to this moment, all tumor cells constantly proliferate, and therefore, in the beginning of the tumor growth, its radius increases exponentially, and the tumor growth curve is thus known. This figure illustrates the distribution of the model variables at the moment when cells at the tumor center have just begun to die, and the expression for tumor radius at this moment can be found analytically, but it is rather cumbersome and therefore not shown. It as well has limits for both infinitesimal and infinitely large levels of nutrient supply. Interestingly, the radius of the quiescent zone does not depend on the nutrient supply level, and here's the simple expression for it. The speed of the tumor growth propagation can be assessed via the mass conservation law, that is, the increase of the tumor volume during the infinitesimal period of time close to the considered moment is provided by the new bone cells in the proliferative region. Like the case of the exponential phase, no explicit expression for tumor radius can be derived for the second stage of tumor growth, and this part of growth curve, however, can be estimated numerically by using the fact that the presence of value of GD with every value in the range from GD to GP yields the expression for values of tumor radius, radius of quiescent zone, and tumor speed for some moment of time during the second stage. Such reasoning results in quite complex ODE that can be solved numerically. The death of tumor cells at the third stage of growth contributes to the deceleration of tumor front propagation, and the distribution of model variables tend to stable profiles depicted here. The staging of glucose distribution function yields quite cumbersome relations between the radii of tumor and its dying and quiescent zones. Another relation between these values is yielded by the mass conservation. The path of the growth curve for the last stage of tumor growth at which dying cells are present can be as well estimated numerically. And this figure compares the tumor growth curves obtained by such semi-analytical methodology with the results of the numerical simulations of the same system, with initial finite width of normal tissue equal to 1 centimeter. As it is seen from the figure, the results produced by two methods are in very nice agreement, and multicolored crosses here denote the boundaries between different growth stages. The limit values for the tumor radius in these moments under infinitesimal and infinitely large values of P are denoted, and the limit overall tumor radius does not fit, but it is approximately 11.34 millimeters. After the obtaining of these results, the dynamics of the full model was studied by numerical simulations under variation of tissue hydraulic conductivity, K over mu, and nutrient supply level P, which is also right here. On the outer boundary, fixed values of normal cell fractions was held, which implies fixed value of external stress. The results will be arranged by decreasing values of K over mu, and visually almost indiscernible tumor growth patterns were produced under the value of K over mu equal to 3 in dimensionless values, which is in the middle of the taken experimental range if we consider it in logarithmic scale. Such high value of hydraulic conductivity led to sufficiently fast smoothing of the solid stress gradients caused by special variations of cell fractions in tissue. This figure shows tumor growth curves obtained numerically in case of hydraulic conductivity value decreased by an order of magnitude, and the growth curves for the already discussed case are also present for comparison as dashed lines. As it is seen from this figure, tumor radii become only slightly lower during the first several dozen days of tumor growth, but eventually they became greater. And the final increase in the tumor sizes was more pronounced with the increase in nutrient supply level. These figures provide two snapshots of distributions of the model variables under P equal to 16 in dimensionless values that allow explaining qualitatively such change in dynamics for the lowest four values of P. The 14th day of tumor growth was the last day when tumor cells had not died yet, and under this value, the fraction of cells within the tumor remains slightly increased due to their proliferation in comparison to their initial value. Since the tumor cells were more densely packed than in case of K of mu equal to 3, while their overall proliferation rates were comparable, tumor had a slightly smaller size during the first days of growth. And the second figure is the stable state. As the movement of the normal tissue practically ceased at this moment, the distribution of fraction of normal cells was almost uniform, close to normal value. Due to the death of tumor cells, their fraction was close to normal only at the interface with the normal tissue and decreased towards the tumor center. Therefore, in order to stop tumor growth, a significantly larger region of dying tumor cells was needed to compensate for the proliferation of cells in the tumor rings, the amounts of which were quite comparable in both cases. That resulted in greater total size of the stable tumor. And note that this figure allows deducing the qualitative pattern of cells and fluid dynamics within a stable tumor. According to the model equations, the direction of cell velocity throughout the tumor in this situation should be opposite to the direction of tumor cell fraction gradient. Thus, the new cells, which appeared on at the outer tumor rim, move towards the necrotic core when the dye turning into fluid, and the fluid moves towards the tumor boundary, where it is used again for cell proliferation. For the largest used value of nutrient supply level, the tumor growth pattern changed qualitatively. Around the 114th day of the tumor growth, the fraction of tumor cells in this tumor center reached the value at which solid stress is minimal. Before this moment, all the tumor cells located close to the tumor center had been pulled towards it. However, upon further decrease of cell fraction in the tumor center, cells situated there began to be pulled away from it. and with further tumor growth, necrotic fluid-filled core, effectively devoid of tumor cells, formed within the tumor center. Accumulation of fluid within the tumor resulted in slower deceleration of its growth. Note that the abrupt declines of in normal size fractions indicate the boundaries of the normal tissue, where it was displayed from its initial position of 1 centimeter. At that, the total volume of normal cells was conserved in every simulation, since they neither proliferated nor died. And note that the normal tissue width therefore gradually decreases. For even lower value of hydraulic conductivity by another order of magnitude, fluid-filled necrotic core formed under every value of nutrient supply level. However, in this case of its lowest values, the presence of the fluid-filled core led to quite moderate increases of maximum tumor radii. For the 3 greatest values of P, tumors grew without saturation for much longer periods of several years, and maximum tumor volumes in these cases increased 5, 8, and 10 times approximately. And the greatest value of P, tumor grew up almost to 10 centimeters in radius. The growth of the 3 largest tumors was eventually ceased due to an effective thinning of the normal tissue region, which could not supply the tumors with sufficient amount of glucose for continued growth. Interestingly, the formation of giant benign tumors in case of low hydraulic conductivity seems to be a physiologically reasonable result. Such situations were observed clinically, for example, in the cases of fibroepithelial tumors, which consist of epithelial and stromal tissues, the latter primarily being made of fibrous extracellular matrix, and in case of fibroids as well, which are benign smooth muscle tumors, which growth is accompanied by ample production of extracellular matrix as well. Other even lower tissue hydraulic conductivity, another effect showed up in the simulations, that is the restriction of tumor growth speed by stress. It happened when tumor cells fraction reached their critical value that corresponded to the critical solid stress at which cell proliferation ceased. As long as such condition held, tumor growth curve was independent of the nutrient supply level. These figures illustrate this via the distributions of model variables for the 2000th day of growth under P equal to 4 and 64. The 2 tumors had equal radii despite obviously different total amount of tumor cells, since only a very small number of them in the tumor reams were actually proliferating. In the stress-restricted growth regime under high values of nutrient supply level, tumor growth accelerated drastically in result of the thinning of the surrounding normal tissue. That is due to the fact that for sufficiently large tumors, a thinner region of normal tissue with fixed values on its boundaries should have steeper profile, which in its turn should yield greater speeds of its movement, as it is defined by the gradient of normal cells fraction. For largest nutrient supply levels, the speed of tumor front propagation after achieving its minimum increased almost 18 times in approximately 8.5 years. This result suggests that significant acceleration of tumor growth may happen in case of benign tumors with low hydraulic conductivity without their progression to malignant phenotype, in particular without alterations in intrinsic proliferation rates of individual tumor cells and without them becoming motile. Such pattern of growth is hard to observe clinically, since it requires long-term observation without intervention. However, at least several cases involving explosive acceleration of growth of benign tumors were reported for cystosarcoma phyllodes, which is a fluid-filled fibroepithelial breast tumor. And note that the stress-induced tumor growth limitation was held until different moments of time, at which the nutrient supply became insufficient to maintain the stress-induced speed limit. The further tumor growth was limited by glucose diffusion, and its speed began to fall down due to the ongoing thinning of the normal tissue. Therefore, the volume of the normal tissue should have an biggest effect on the pattern of tumor growth. This is illustrated here via the tumor growth curves for the lowest hydraulic conductivity and average nutrient supply level under the right initial radius of normal tissue region. High normal tissue volumes initially restrict tumor growth by solid stress to a greater extent, but eventually produced longer growing tumors with greater maximum volumes. Okay, I suppose that I don't have time to shortly repeat what has been achieved in this work, but I will state that the key task that will be focused on with the use of the developed version of the presented model is the optimization of various types of long-term tumor treatments associated with the delivery of drugs via intravenous injections. And due to this type of the task, it is crucial to keep the numerical cost of the model as low as possible, because you need to conduct a lot of simulations in order to get result in such types of work. And the most crucial model drawback that will be certainly included is the consideration. The drawback is the neglect of circumferential stress, which arises as the normal tissue stretches due to the growth of the tumor within it. And it will be certainly accounted for, but it's not a trivial task to keeping the numerical cost low. So thank you for your attention. The results are published, and you can find all the details of my work in my recent article. Thank you.
Thank you very much, Maxim, for this very interesting and important presentation. It was quite intense, and sometimes there were some problems with connection, I think. So I think that some points.
I'm sorry. I'm now on vacation in Egypt, but I decided to still make it up.
Okay.
So the internet in this country is very poor.
Well, I'd like to recall our listeners that they can ask questions either by chat or raising hand, and we have several minutes for some questions. So please, you can ask. Could you please explain us? Maybe I missed. Or maybe summarize. So you introduced solid stress in the model. So could you please explain in a simple way how this solid stress acts on tumor growth? Does it accelerate it, or does it just decelerate it?
No, the solid stress, if we account for it, it can only decelerate the tumor proliferation rate because it acts. It is, roughly speaking, it is difficult for a single cell to proliferate when the stress acts upon its membrane, because it has to divide into two cells which are repelled from each other by some intercellular mechanisms. So it has to effectively increase the volume it occupies. And it is harder to do it when the solid stress acts on it. So the cells have some biomechanical cues that they sense, and that slow down their proliferation. So that is essentially the process that holds the normal tissue from overproliferation. And this is a bit damaged in cancer cells, so their threshold is lower, but still they cannot divide it infinitely large solid stress. So there is a certain threshold where they cannot divide at all, and there is a certain slowing down when you apply the greater and greater solid pressure.
Yes, right. Thank you. It's a good explanation. So could you estimate somehow in the model, not in the just real, of course, biological experiments, but in the model, the importance of this solid stress, so how much it can influence the rate of tumor growth?
Well, I have done it in the latest part. This is namely the tumor growth patterns, which are largely influenced by tumor solid stress, by solid stress. So it restricts tumor growth in the beginning, but then when it doesn't restrict the growth no longer, the tumor growth acceleration happens. So that's a problem which interestingly has been noticed in clinics not once and not twice, and for certain types of benign tumors. I underline benign tumors that don't switch to the malignant phenotype.
Yes, okay, thank you. So Alberto Danufrio has a question. So Lilia, please give right to Alberto to ask question. Switch his microphone. Lilia, do you hear me? Yes, okay, Alberto, you can ask. You can switch your microphone and you can ask question.
No, sorry, I was not finding the icon of the microphone. very interesting presentation because not enough research effort is devoted in mathematical oncology to benign tumors. I'm very interested to understand a little bit better the physical reason why very low hydraulic conductivity helps the velocity of growth of tumor and reaching also the giant benign tumor steady state. Thank you very much.
Well, as far as I understand, you want me to explain this a little bit more detail. The reason is that under very low hydraulic conductivity, the solid stress does not equalize fast. So only within the significantly low values can tumor cells reach their critical value of density within the tissue. So this is the crucial moment. Under higher values of hydraulic conductivity, the tumor cells did not reach this value. Of course, I use only a very simple function of dependence of tumor cell proliferation on the solid stress. In reality, certainly with the increase of tumor cell density, their proliferation rate will slow down quite monotonically when the density of tumor cells will increase. However, in this model, only under very low values of hydraulic conductivity, the solid stress cannot equalize at the time scales sufficient in comparison with the scale of tumor growth. So they reach this critical value of density. And these cells not proliferate. Only a very small amount of cells near the tumor rim where their density is slightly lower than this critical value, which cannot be seen at such magnification.
Well, thank you very much for this response. I think that we have to,
thank you.
To stop now the discussion. Thank you again for this very interesting presentation. And we will move now to our next speaker, Professor Wertheim. You can share your screen. Yes, we see your screen.
Yeah, can you see it?
Yeah, we see it, not the
hello?
Not full screen, it will be a little bit larger.
Can you hear me?
Yes, yes, we hear you and we see your screen.
Okay,
so,
yeah.
Yeah, now we see, yes, now it's full screen. Okay, thank you very much. So it's a pleasure for me to introduce our next speaker, Professor Wertheim from the University of Sheffield. And the title of neuroblastoma. So please, you can start your presentation. We are listening to you.
Oh, yeah. So thanks for inviting me. And my talk is probably a bit different from what you're used to in this workshop because it's actually computational modeling. Did I lose the screen? Yeah, because my talk is actually computational, not mathematical. And I'll talk about the so I will talk about to my best knowledge, the very first multicellular model of neuroblastoma. Today we have four objectives. A, I will introduce you to the pre-MAGE project. B, I'll talk about the scale separation strategy used in the project. And C, I'll introduce you to the very first multicellular model of neuroblastoma. And finally, I'll talk about the calibration strategy used to parameterize the model. So first, I'll tell you the vision, what we're trying to achieve and how we're doing it in this pre-MAGE project. And actually, someone has already done it for me.
Well, I'm sorry, is it animation accompanied by some comments or it is silent? Because I don't hear anything now.
And just to confirm, you can hear me, right?
Yes, we hear you, but in fact, during this animation,
Yep, great. Yep. So yeah,
explain
so
something because the sound was not there.
Okay, dear. Yeah, so I mean, so if the sound of the video was missing, then you can just check it out later. But to sum up the pre-image project, we are trying to build a decision support system for the clinical management of malignant solid tumors. Think of it as a pipeline. It will take images, process it, segment it, combine it with other biomarkers such as genomic profiles in order to parameterize multi-scale models covering different scales from organ to molecular, run multi-scale high-throughput simulations and use machine learning techniques to extract insights from the simulation results. Moving on to the second part of this presentation about scale separation. We are doing this project within the context of neuroblastoma. Later, we'll also look at diffuse intrinsic pontine glioma, but right now our focus is neuroblastoma. It's a pediatric cancer. It is the most common extracranial solid tumor in children, and is responsible for 15% of mortality in that population. And the primary tumor is usually found in the adrenal medulla. Where does it come from? It comes from the neural crest, which is a transient structure in the embryo. The cells in the crest migrate to different parts of the embryo to differentiate into different cell types, including the sympathetic nervous system. When it's impaired, it will lead to cancer, such as neuroblastoma. The adrenal medulla is linked to the sympathetic nervous system. A major point about neuroblastoma is that it is heterogeneous in many ways. In the benign case, you don't need to do anything. Just observe the patient and the tumor will shrink spontaneously. But at the opposite end of the spectrum, they can develop drug resistance and metastasis, even after multimodal treatment. A key biomarker is amplification of the myosin gene. And it is usually a bad thing. When you have myosin amplification, it's usually high risk, and the survival rate is below 50%. Here, I show you the hallmarks of cancer. There are many, such as sustained proliferation, resistance of apoptosis, modification of microenvironment. These biological phenomena span 9 orders of magnitude in time and in space. My colleagues in Italy have looked at this problem. They actually do literature review and wrote down the scales, and their conclusion is 9 orders of magnitude. And I can tell you it's impossible to write down a model, say, using partial differential equations to cover 9 orders of magnitude for 3 reasons. First, experimental resolution. Our measurements are limited, and as a result, there are certain areas in those 9 orders of magnitude where you can't take measurements. Without data, you can't parameterize the model. Second is model complexity. Even if we could get all the data for parameterization for all 9 orders of magnitude, think about the complexity of the PDE model. And by extension, it will be expensive to solve it. As a result, we separated the scales. We picked the most critical parts of the scales about the biology. We've decided on the whole tumor scale and patches of the whole tumor, and that's a tissue scale. And within this tissue scale, we also look at each cell individually. My colleagues in Zaragoza use partial differential equations to model the tumor scale. At the University of Sheffield, we combined discrete agents, continuous automaton, and center-based mechanical model to study multicellular interactions in patches of the tumor. And each of the cells is informed by a subcellular model developed by a pharmaceutical company called Chemotargets, and they use machine learning to do it. And next, I'll look at the major contribution of my university, which is the tissue scale multicellular model of neuroblastoma. To the best of my knowledge, no one has built multicellular models for this cancer type. Our model has 3 components. The first part is a continuous automaton. It voxelates the tumor microenvironment into small cubes. It uses this grid to track the spatial distributions of cells and matrix over time. Associated with the automaton are the abundance levels of nutrients, including oxygen and chemotherapy, as well as the general level of inflammation, which is just a static parameter. Populating the continuous automaton are discrete software agents. I have 2 populations, neuroblastic cells and Schwann cells. I should point out that the 2 cell populations are the major components of neuroblastoma. The neuroblastic cells are the cancer cells, and the Schwann cells are the stroma cells making up the volume, and they interact. For example, Schwann cells promote differentiation and apoptosis in neuroblastic cells, and in the opposite direction, neuroblastic cells promote proliferation in Schwann cells. So it's a negative feedback loop. To model the interactions, I use a device called 3D von Neumann neighborhood. If I want to study one cell, I'll identify the voxel it is in, as well as the voxels immediately adjacent to it. Then I'll count the relative abundance of Schwann cells and compare it with the abundance of neuroblastic cells. This comparison will tell me which interaction is dominant. Each agent is characterized by attributes, including mutation status, such as myosin amplification, the activities of other genes, and the status of its DNA. Is it damaged, or are its telomeres too short? Broadly speaking, each agent can do 2 things. They can grow and they can die. But under the hood, there are more elaborate regulatory mechanisms which allow the agents to respond to the microenvironment and each other. Obviously, the cells are physical objects. They can overlap, and when they do, that's called contact inhibition, and the cells cannot cell cycle, cannot grow. And I use a center-based mechanical model to resolve cell-cell overlap. Tumor progression is a dynamic process. To simulate this process, I split it into smaller time steps, and I used the model I just showed you to predict the events occurring in each time step. In my model, each time step is 1 hour of the patient's life. And during this time step, I use the model to do the following sequentially. I look at each cell individually, update its attributes, let it grow, let it die. Then I resolve the overlap using the mechanical model. And finally, I consider all the cells collectively as a population, and on that basis, modify the tumor microenvironment, such as consuming oxygen. So in more detail, we start with the cells individually. Each cell will sense its microenvironment. How much oxygen is available? Is that chemotherapy? And in the case of a neuroblastic cells, is it being influenced by Schwann cells? And more generally, is the neighborhood too crowded? Is there contact inhibition? And response, the cell will modify its behavior. Recall that Schwann cell can promote differentiation in neuroplastic cells. And I can tell you, when you can get the neuroblastic cancer cells to differentiate into neurons, the tumor will become benign. It won't be dangerous at all. And that's because more differentiated cells are less likely to undergo cell cycling and proliferate. And there are also other contributing factors to proliferation rate, such as the abundance of oxygen and cell density. When a cell divides, the telomeres capping its chromosomes will shorten, and this will induce damages in the DNA. There are other contributing factors to DNA damage, such as hypoxia, lack of oxygen, or obviously chemotherapy. When the cell is too damaged, it will die by apoptosis or necrosis. And the corpse, the carcass, will be removed by the immune system. I have told you that the cells can overlap, and when that happens, it's contact inhibition, which will inhibit cell cycling. And to model cell-cell repulsion to resolve the overlap, I use a center-based mechanical model. As a cell gets bigger in the cell cycle, it overlaps more. And I use a simple equation to calculate the extent of overlap. And using the total overlap, I just plug it into a linear force law to calculate the net force acting on that cell. And I plug the force into an overdamped version of Newton's second law to update its displacement, update its coordinates. And I iterate these steps until I get convergence. This is similar to molecular dynamics simulation. And finally, to finish the time step, I consider the cells collectively in order to modify the microenvironment. If there are a lot of cells, then oxygen and nutrients will be consumed at a high rate, and the cells can remodel the vasculature and produce matrix. To sum it up, tumor growth is a dynamic simulation. I split it into smaller time steps. In each time step, I let each agent sense the microenvironment, modify its behavior, and update its attributes. After evaluating every single cell, I resolve their overlap using the mechanical model. And to finish off the time step, I consider the cells collectively in order to update the microenvironment, such as the abundance of oxygen. Now we'll look at the last objective of today's presentation. How did I calibrate this model? This is a major challenge for me. It's a complex system model, and I fixed most of the parameters using literature values, but there's still a lack of data, and there are 20 fitting parameters. I use a technique called Latin hypercube sampling to obtain 3,000 parametric combinations in the 20-dimensional parametric space. And then I set up a selection process like a tournament. I started with 3,000 candidates, the parametric combinations, and I make them go through a series of 6 studies. In each study, I compare the simulation results with experiments and clinical observations to eliminate the unrealistic parameters until I found the best set of parametric values. And guess what this reminded me of? I'm not sure if you have watched it yet, but there's a hit series called Squid Game on Netflix, and it's a similar idea. You start with a bunch of candidates, make them do tasks, and then get rid of the unfit ones. In the first round, I consider a study from the 1970s. It's a very general study about the growth kinetics of neuroblastoma cells, which were cultivated in a growth medium, and the cell population was tracked over time. I used each parametric combination to simulate tumor growth, and I fitted the results to the data shown on the left to calculate the residual sum of squares. This allowed me to eliminate the bottom 2,000 combinations. The remaining 1,000 entered the next round. This time, I used another modeling study as my source. They have done the hard work for me, and they did their own data fitting. And in the paper, the oxygen level at which 50% of cells would die because of hypoxia is reported. I used each of the 1,000 remaining combinations to simulate hypoxia, and the aim is to see how close the death rate is to 50%. Considering the 2 studies holistically, I eliminated most combinations and only 50 remained. And they entered the 3rd round. And this time, I look at something more specific. So as you can see, I went from a very general case about growth kinetics and hypoxia to a more specific case about how neuroblastoma cells and Schwann cells influence each other. And that's because where fewer candidates left, they are the best ones, so we need to be more discriminatory. In that experimental study, neuroblastic and Schwann cells were co-cultivated, and the fractions of proliferating neuroblasts and Schwann cells were tracked over time, as well as the fraction of apoptotic neuroblasts. I used each of the remaining 50 parametric combinations to simulate the crosstalk between the 2 cell populations. I fitted the results to what I show you on the left, and I picked the top 10 combinations. The next study is actually based on a set of clinical observations. So we moved from relatively coarse experimental data to more refined clinical observations. So far, I've been talking about neuroblastoma, but strictly speaking, that's a bit dishonest. What I actually mean is neuroblastic tumors, of which neuroblastoma is one type. And in total, there are 5 histologies. And a tumor has three major outcomes. It can regress spontaneously, it can differentiate into harmless neurons, and it may become malignant. And the different histologies are associated with different tendency towards the three outcomes. I used each of the remaining 10 combinations to predict the clinical outcomes for different histology types, and only 4 of the remaining combinations gave results resembling the observations by clinicians. Then I considered another clinical study. In this study, they look at tumors with different combinations of mutations, although myosin is not amplified, and they look at the clinical outcome. Similar to before, I used each candidate to predict the clinical outcome and are associated with different mutations, and 3 combinations remained. And finally, I used the same set of data, but this time I considered the myosin amplified tumors, and the results are not conclusive. And that's why I took a step back. The 3 remaining parametric combinations, I look at all 6 studies holistically, and I pick candidate 564, which doesn't mean anything to you, but it's just to remind you, it simply is a set of 20 parametric values. So I know you may wonder, wow, I'm looking at millions of cells of agents, and I'm simulating their evolution in the course of 4 months. The model is stochastic, so I need to repeat the simulations over and over again. So it's so expensive. How did you do it, Ken? And I use high performance computing. I ran the simulations on GPUs enabled by a software called Flame GPU developed at our university. In short, it maps model descriptions to optimized CUDA code. Recently, our colleagues released Flame GPU 2. This software allows you to focus on the building of models without worrying about CUDA programming and GPU optimization. You can easily simulate millions of agents. It allows visualization in real time, and there are Python and C++ APIs for describing the model. Apart from this project, it has been used for immunology and logistics. And some statistics about the computational time. I used my colleague and I used 4 GPUs and to implement 300 time steps of my model. It took 5 to 10 minutes. And the 6 studies I just reported took 40 days. Without GPUs, you cannot do it. Now that the model has been calibrated, my next step is to introduce clones of cells with different mutations and simulate tumor progression. In short, I want to see how they compete with each other. And by modeling clonal evolution, I want to answer 2 questions. What makes some neuroblastoma regress and differentiate? And second, how can we manipulate this knowledge? How can we use it to induce regression and differentiation, which is what we want? To sum it up, the PRIMAGE project aims to build a decision support system for the clinical management of malignant solid tumors. Cancer hallmarks span 9 orders of magnitude in space and time. Because of limits posed by experimental resolutions, model complexity, and computational costs, it is necessary to separate the scales and orchestrate information flow between them. At the University of Sheffield, we built, to the best of our knowledge, the very first multicellular model of neuroblastoma by integrating a continuous automaton, discrete agents, and a center-based mechanical model. The multicellular model has 20 fitting parameters. We calibrated them in a Squid Game style tournament. we use Latin hypercube sampling to generate 3,000 combinations of parametric of parameters, aggregated experimental and clinical data. And we use the data to design 6 in silico studies performed on GPUs to assess and select the combinations. And sorry about the explainer video. After the presentation, I'll send you a link so that you can watch it in your own time. Thank you.
Yes, thank you very much for this very interesting presentation. And we have some time for questions. And waiting for questions from our listeners, I would like to ask you that as far as I understand, you use soft sphere model for cells, right?
Yeah.
Of latest soft sphere model. And what I did not exactly understand, or maybe I missed something, do you consider intracellular regulation in your model?
Yes.
Because your cells, sorry, you say that they can die or they can proliferate. So how it is determined? Is it the level of oxygen that determines the cell fate?
Yeah, that's a very good question. Yeah. So when I put this presentation together at a high level of abstraction, and all those the major events are cell division and cell death, but they're regulated by an elaborate intracellular signaling network. So for example, I have MAPK RAS pathway, which is related to cell death, and I have P53 and P73, which are related to apoptosis. And this elaborate network of intracellular events will link the microenvironment, such as a lack of oxygen, to the final outcome, which is cell cycling and dying. For example, a lack of oxygen, hypoxia, will trigger HIF. And HIF will also trigger P53 to induce cell death. And I hope that answers your question.
Yes, thank you very much. There are sometimes some problems with sound. It's a little bit but this intracellular regulation, do you consider it? For example, there are different approaches to model intracellular regulation. It can be, for example, ordinary differential equations. This is what you do, or you do something different?
Well, I would say it's closer to Boolean network modeling.
Boolean network, okay.
Yeah, but it's not exactly a full-blown Boolean network. It's more like conditional statement. So for each one, I use Bernoulli trials to determine the value. So I will have a probability for P53 activity level. And so if there's not enough oxygen, then I pass the first conditional. After that, I do a Bernoulli trial using the activity level of P53 as the probability of success, and I determine the outcome of the Bernoulli trial. Yeah, so I guess this is quite different from the rest of the talks because you mostly look at differential equations, but this one is agent-based.
It's of course it's different from other talks, but it's in the same framework, and we understand in this audience very well because many people from this audience also use similar methods. And for example, Anas Bushnita from the audience asked a question through the chat and comments. He writes, interesting approach for parameter selection. Does the model account for the migration and influx of neural crest cells to the site of the tumor?
Cell migration. Yes.
Yes, migration and influx.
Oh, yeah. My boundary conditions are provided by my colleagues from Zaragoza. So imagine this is what I have here. This lump is the multicellular model at Sheffield, and it is a part of it is a patch in the tumor scale model built by my colleagues. and they provide me with a displacement field as a boundary condition. So if my cells go beyond a certain boundary, there will be a force field to push it back into the lump.
Right. Okay. And Anas continues his question. What do you consider as the initial condition for your simulations?
Initial condition. Let me think. A few things. So the most important is the cell count. So how the relative abundance of Schwann cells and neuroblastic cells, because as I mentioned, their interactions are very important. So the relative populations of the two cell types, the abundance of oxygen level, and that is related to the state of vasculature, which is provided by our collaborators. And we also have the availability of chemotherapy, chemotherapeutic drugs. And we also have cellularity, because in our lump, we don't just have the cells. We also have extracellular matrix, which is modeled as a concentration field. And if a voxel is full of matrix, then the cell cannot divide because there is contact inhibition. And also the mutation profile, because I mentioned that we have myosin amplification and other mutations. And at the beginning, we need to specify whether the cells have myosin amplification or not. So those are the major ones.
Right. Thank you very much. It's in fact, this is what we call hybrid models, where we have combination of the cell-based model and intracellular regulation, extracellular matrix. We did it for other types of tumor. And Anastasia Mazokhina from our audience asks, does this approach is applicable to the other kind of tumors, and what will change in that case?
I suppose the answer is yes and no. It's yes and no. So we are building this decision support system for malignant solid tumor, and we are exemplifying it using neuroblastoma and later on diffuse intrinsic pontine glioma. So so obviously it will be useful for those two types of tumor. But if we change our parameters, if we change the intracellular network, we can use the software to describe other solid tumors, such as pancreatic cancer. But what we cannot do is to study leukemia, for example, because that's not solid tumor, that's blood cancer. So I guess the answer is it depends. If it is solid tumor, then we can adapt our software to study other tumor types. But if it's blood cancer, then we cannot. And also just now in the chat, I sent out 2 links for the explainer video. It really is a shame. I don't know why the sound was gone.
No problem. Thank you very much. And just to finish our discussion, you mentioned leukemia. I would like to say that we used similar models. We call it hybrid models to model leukemia and multiple myeloma. It's another type of blood cancer. But unfortunately, we have to finish this discussion. Thank you very much for the very interesting presentation. And we will move to our next